ANRF-PAIR project, ANRF-PAIR SAKSHAM project at IIT Indore. The ANRF-PAIR SAKSHAM project, led by IIT Indore through its hub spoke model, aims to create strong, impactful research ecosystem. So today, in this webinar, we are joined by our speaker, Professor Avinash Sonavane. Professor Sonavane is a professor in the Department of Biosciences and Biomedical Engineering at IIT Indore. Professor Sonavane has earned his PhD degree in Human Medicine from University of Marburg, Germany. Also, Professor Sonavane has a postdoctoral fellowship from prestigious European Molecular Biology Laboratory, Germany, Max Planck Institute for Infection Biology, again from Germany, Department of Medicine, University of Florida, USA. He also worked as a research scientist at Department of Bioscience, University of Oslo, Norway. He is an Humboldt visiting professor at Leibniz Lung Center, Germany. In past, before joining IIT Indore in 2018, Professor Sonavane briefly served KIIT Bhubaneswar. He is also a Humboldt Fellow, EMBO Fellow, JSPS Fellow, Swiss National Foundation Fellow, and Swedish Institute Fellow. His research focuses on tuberculosis pathogenesis, host-pathogen interactions, antimicrobial therapeutics, targeted drug delivery, and novel therapies for leukemia. He has published over 90 research articles, secured multiple patents, transferred 2 technologies to biopharma companies, and received several accolades and awards, including SERB Technology Transfer Award, National Bio Entrepreneurship Award, and ICMR Major General Sahib Singh Shoki Award. So with this brief introduction, I invite Professor Sonavane to enlighten us with his work, and I'll be guiding this and hosting this webinar throughout this time of 45 minutes. The webinar after sir finishes, then we'll have a quick question answer series as well. Over to you, sir, for the webinar. Professor Sonavane, I invite you for the presentation.
Okay, am I audible?
Yes, sir. Yes, sir, you are audible, sir.
Okay. So thank you, Dr. Ankur, for the nice introduction. First of all, I would like to thank. Okay, let me share my presentation first. So can you see my presentation?
Yes, sir. It is visible.
Okay, thank you. Yeah, so thank you very much. First of all, I would like to thank our R&D section and PAIR coordinating committee for their kind invitation. So today I'm going to talk about the healthcare innovations and some technologies that have been developed or under development in our laboratory. So today I'm particularly going to talk about, in the first part, I will talk about some therapeutic drugs that we have developed in our laboratory. And these drugs we found that can improve the treatment of primary as well as relapsed leukemia, which is a type of blood cancer. And then in the second part, I will talk about the 2 diagnostic point-of-care diagnostic kits that are under development in collaboration with our collaborator from Japan and Denmark for the diagnosis of active as well as multi-drug resistant tuberculosis. So before I start talking about these innovations, I think first we need to understand what is.
Sir, just a minute, sir. Can you just make it in a full screen, sir? It is not in a full screen. Yeah.
Okay.
Perfect.
Yeah. Yeah. So before I start to talk about these technologies, let us first understand what is innovation. By and large, we all think that innovation is nothing but to discover or to develop something new, and I think that is not true. Innovation is not just to discover or to make something new, but it can also be that if there is some technology or some product is available, so can we improve this technology by identifying the limitations in that particular technology or in that product and to try to make it better? So this is not like one way of innovation. And another way of innovation is also that if some product has application to a particular disease, treatment or therapeutics or whatever, so can we use that technology or that product for treatment or for other applications? So that is called as repurposing of technology. So these are not like various ways of innovation. So innovation, like it is a ladder that like we need to climb, and at each stage we need to face different challenges. So this as you can see, like this is a typical flow chart or the channel of this innovation. So the first step of this innovation is to do some basic science or some basic research. So if you want to develop some product or some technology, we need to do some fundamental work. Okay, so identify that how this product can be developed, what are the features that this product need to have. So first we need to do some basic research. And in most of the cases what happens that most of these researchers, they like want to publish their basic findings. But if some component has some innovative components, like that leads to the next stage, that is called as the lead molecule. So from a basket of like different molecules, you come up with some lead molecules, and then some of these lead molecules can be patented if there is some innovative compounds. So and then this patent compound, some of these patents, it may lead to development of some prototypes, and then finally these prototypes are then goes to the next level, that is like to the some product development or some technologies. So this is called as like bench to bedside. So the basic findings of the research is called as the bench, and if that basic finding reaches to the development of some product, then it is called as the bedside. So as you can see here, it is in basket. So when you are doing this basic science, you will come up with like thousands of lead molecules, thousands of materials, many different technologies, or you may develop many different these devices. But these all these innovations of the these prototypes, it goes through like various filtering process, and then maybe like 0.1 or maybe even then less than 1% of these molecules or these technologies, it leads to the products. And so just to give an overview that what our lab does. So we work on these two diseases. One is tuberculosis, and in tuberculosis we work on different domains. So we are trying to understand the mechanisms of drug resistance and how this drug resistance, it modulates the pathogenesis of this tuberculosis infection in human and in animal models. Uh, the other domain we work on is to understand the epigenomics of the macrophages and stem cells, and how these epigenetic modifications that help in the progression of tuberculosis disease. So these are the two components. Like we do a basic research, and then on translational aspect, we work on development of some novel therapeutic molecules for tuberculosis treatment, particularly the multi-drug resistant tuberculosis. So we are identifying or we are synthesizing some therapeutic molecules or some small molecules that can target this multi-drug resistance in tuberculosis. And we also work on developing some point of care diagnostics for tuberculosis. So today I'm going to talk about or show you some data on the two diagnostic the kits which are under development. The other area we work on is leukemia, or it is there are different types of leukemia: myeloid leukemia, acute lymphatic leukemia. These are the different types of blood cancer. So we mainly focus on acute lymphatic leukemia. So this cancer is more predominant in children below 15 years of age. So in leukemia, again, we work on different areas. One is the enzyme engineering. So there are enzymes used for the treatment of leukemia. However, these enzymes, they have several limitations, drawbacks. So we are trying to improve this therapeutic efficacy of these enzymes by an enzyme engineering approach. The other thing we are working on is to do some modifications, particularly different types of chemical modifications in this engineered enzyme, and trying to further improve their therapeutic the efficacy. We also work on the one of the major problem that these leukemic patients they suffer when they take these drugs is the blood coagulation, or that is also called as the thromboembolism. So we're trying to understand the molecular mechanisms of the blood coagulation in ALL patients. So these are the different areas we are working on. So in leukemia, today I'm going to talk about one of these enzymes which we have engineered in our laboratory to improve this ALL therapy. Sorry. Yeah. So since like many students are attending this webinar, so first I would like to like discuss that what is required for the innovative research. So the first and the most important thing is to identify the gaps. So what does that mean? Identify the gaps is, for example, if there is some disease which has, I don't know, like lacks some therapeutics or some vaccines or some diagnostics. So we need to understand, like how to, if there is no therapeutic drug available, so can we develop some new therapeutics? Or if there is no proper diagnostic method available, so can we develop some diagnostics? Or if there are already some therapeutics or diagnostics are available, so are there any limitations or drawbacks of this existing one? So how can we improve these therapeutics or the diagnostics? So we need to identify those gaps and work on those gaps. Then second thing is the feasibility, and then we need to ask this appropriate question, because ultimately these technologies will be taken up by the industries. Okay, so what is the feasibility of this product? So for example, if you are developing let's say therapeutics for treating some disease, so is that disease really predominant in that country or in that particular region? Because if you are developing a drug or vaccine, and if that disease doesn't exist in that region, so it makes no like, so its feasibility becomes a problem. So we need to ask this appropriate question and also think that whether development of some technologies or the products has some feasibilities or not. Then we need to work on the idea. So how to develop, for example, like if you want to develop some device, so how to make this device more user friendly, cost effective, specific. So like we need to identify those limitations, and then we need to work on the ideas which will overcome these challenges. And of course, like development of any product, any technology, it needs a continuous like funding. So we also like need to like collaborate with different institutes, industries, or maybe the funding agencies, and like try to generate some funds which can support your work. And the most important thing is that you need to find right industrial partners to take your innovations or your technologies to the market. Because without industrial support or the participation, it becomes very challenging to take your technologies to the market. And then of course you need to have confidence in your innovation, because for example, for diagnosis of a particular disease, there are many different types of like diagnostic methods are available. So how your innovation is better than these existing ones? So you need to have very innovative idea, and that will like you need to have confidence in your whatever product you are developing. And of course it needs a lot of patience and perseverance, because innovation does not happen overnight. It takes like few years. I'm just giving example of the therapeutic molecule. As you can see here, to develop one therapeutic molecule, it takes between 10 to 15 years, because like it includes a different steps. For example, like the first step is when you want to develop a therapeutics, first you need to identify the target, then do some in vitro, in vivo studies, then next one is the preclinical studies. You need to like go through this lot of regulatory hurdles, then clinical trials, then like regulatory approach, and then it comes to the market and commercialization. So this whole process takes like between 10 to 15 years. So one need to have like patience and perseverance while taking up these challenging things. So how to overcome these challenges? One is, of course, you need to like have focus to develop something, and then you need a multidisciplinary approach. For example, if you want to develop a drug, to like then you need, if you need a chemist, you need a physicist, you need pharmacologist, you need clinicians. Okay, so you need to have like collaboration or the teamwork to develop a product which has a wide application. And of course, as I mentioned before, you need to collaborate with the industry. So as you can see, like the development of product or innovation, like it's a ladder. First there is an ideation phase, then you need to plan it, then you need a teamwork, and then only like you can reach to the top of this ladder. So just before, like there are many different therapeutic strategies are used nowadays. Like AI, like we all know what is AI. So AI has like large, great application in developing these drugs, target identification, studying its mechanism. Then of course, like you need to have different models which will test the efficacy of your drug. So it can be done in vitro models, or now you can also develop the organoids. Now there is 3D bioprinting. So these are different ways by which the efficacy of your molecules can be tested. You might have heard about this gene therapy. If there is some defective gene or particular gene which like is responsible for causing that disease, you can edit this gene by technology called as CRISPR-Cas. So this is called as the gene therapies, like to treat these certain diseases. And then you can also like develop new formulations, new drugs, deliver them to the target sites. You can use some smart nanomaterials to deliver these drugs to the target site. So these are the various strategies which like are required to develop a therapeutics. So just to give an example, these two diseases like we are working on, and these are very prominent diseases, or very like global challenging diseases. Tuberculosis, as you can see, like about 10 million cases are identified or suffering from TB. 1.3 million people they die from tuberculosis every year, and India is like one of the highest burden country of TB. Like 1/4 of these TB cases they are identified in India. And same is the case with cancer. As you can see, it's a global burden. Like a lot of people, like millions of people, they are dying from different types of cancer every year. So coming back to the leukemia, so as I said, we are working on developing these therapeutics for acute lymphatic leukemia, which is a blood cancer. And how this disease is caused? So normally, like white blood cells, they are very important for to fight these infections. But sometimes these white blood cells or WBCs they get transformed into immature or the cancerous WBCs, and then they multiply very fast, and then they number the healthy WBCs. So among different types of leukemia, the ALL is a very predominant this cancer. As you can see, it accounts for almost 70 to 80% of ALL cases. And just in children below 15 years of age, this ALL it represents almost 23% of all cancers diagnosed in children. So you can see that like how predominant this disease is in the children. In India, about 50,000 new ALL cases are identified. And the important thing is that in ALL chemotherapy, this asparaginase enzyme is used in the chemotherapy. And a lot of studies has shown that the inclusion of this enzyme in ALL chemotherapy, it increases the survival of these ALL patients to more than 80%. And there is no substitute for this enzyme. So these are the four like technologies that has been developed in or under development in our lab. We have developed these two drug molecules. One can be used to treat this primary leukemia. Other molecule can be used for to treat this relapse leukemia. Then we have developed this rapid and non-invasive tuberculosis kit. And then we also developed one enzyme formulation which can reduce the acrylamide, which is a carcinogen in processed food products. And this formulation can reduce the cancer development in when there is a consumption of this processed food. So coming back to this ALL, that this L-asparaginase enzyme is very important, or is an important component of this ALL chemotherapy. As you can see, there are different stages of this chemotherapy. One is induction, consolidation, maintenance, reinduction, or it is also called as intensification in high-risk ALL patients. And as you can see, in all stages, that is in induction or consolidation or maintenance, this asparaginase enzyme is used. So how this enzyme, it acts as a therapeutic agent. So normally what happens in blood, in normal cells, so this enzyme is responsible for conversion of this asparagine into aspartate. So in normal cells, the aspartate which is present in the blood is taken up by these normal cells, and these cells, they have one enzyme called as asparagine synthetase. So this enzyme, when this aspartate goes inside the cell, this aspartate is converted into asparagine by this asparagine synthetase enzyme, and this asparagine is used for the protein synthesis, and as a result, these cells, they multiply and they survive. The another source is that our blood also contains this asparagine amino acid. So these normal cells, they directly take this asparagine from the blood and use this asparagine for protein synthesis, and these cells, they survive. But when the cells get transformed into cancerous cells, or these are also called as blast cells. So the aspartate which is taken up by these cells, but these cancerous cells, they lack this enzyme called as asparagine synthetase. So even if they take this aspartate from the blood, this aspartate is not converted into asparagine. So there is no asparagine for protein synthesis. So these cancerous cells, they are dependent on the asparagine which is present in the blood. So then they use this asparagine for the protein synthesis, and they survive. But when this asparaginase enzyme is given, this asparaginase enzyme, it depletes this asparagine which is present in the blood. So these cancer cells, they are not able to convert this aspartate into asparagine because they lack this enzyme. And when this asparaginase is given, it also depletes this asparagine which is present in the blood. So these cancer cells, they are devoid of this asparagine. So there is no protein synthesis, and these cells, they undergo apoptosis, and they die. So currently there are many different types of this asparaginase drugs are available in the market by like several many different companies, particularly from Europe, Japan, and North America. They are manufacturing these L-asparaginase drug with these different these brand names. However, these drugs, they have several limitations. One is it has very low activity. As a result, the patient has to take repeated doses of this enzyme, and that causes toxicity to the vital organ. These drugs, they are not effective in the treatment of relapse leukemia, and they also cause allergic or hypersensitivity reactions. And another major problem is that this enzyme is derived from bacteria, and it is given to human. And because of this immunogenic nature of this enzyme, the patient body, it produces antibodies against this enzyme, and that causes the hypersensitivity reactions. And because of this production of this enzyme, antibodies, it neutralizes this enzyme, and it reduces its therapeutic efficacy. Another major drawback of this enzyme is that it also has this glutaminase side activity, and this glutaminase activity is responsible for neurotoxicity, or which is also called as a brain toxicity. So because of these various limitations, it leads to different side effects. Like it causes inflammation in the pancreas, toxicity to liver toxicity, brain toxicity, thromboembolism, or that is a blood clotting. So these are the several side effects of this drug. And as I said, this is an immunogenic protein derived from the bacteria, and it is given to human. So when this asparaginase is given to ALL patients, this patient, their body, they recognize this protein as a foreign substance and produces antibodies against this drug. And but when there is a second administration, so this ALL patient, they already have these antibodies. So when this enzyme is given again, this pre-existing antibodies, they bind to this enzyme and neutralizes this enzyme. So this enzyme is not able to achieve its therapeutic efficacy, or not able to kill these cancerous cells, and as a result, these cancerous cells, they multiply and cause a leukemia. So then how to overcome this solution? So we need to develop a new asparaginase molecule which will be more stable. It will have reduced glutaminase activity, which is responsible for the brain toxicity, reduce immunogenicity, so there is no antibody production in the patient body. It should not have any toxicity on the vital organs, and it should also improve the treatment of relapse ALL. So to achieve this, we engineered this enzyme by protein engineering approach. So we did like a lot of modifications in this enzyme to overcome those side effects, which I have already like mentioned in the previous slide. So how we did? So we created more than 12,000 changes in this protein, and then we screened these proteins from 12,000, and we look for these properties. So each variant was checked for its activity, glutaminase activity, immunogenicity, cytotoxicity, then organ toxicity. So from 12,000, then we found 5,000 lead molecules. Then we again screened this 5,000, and then we got 1,000 lead molecule. And then finally, we found that there is one variant, or we call it as a mutant. We named it as M-ASPAR, which has these properties, that it showed high enzyme activity, low glutaminase activity, low immunogenicity, no toxicity, and more cytotoxic towards the leukemic cells. So I will not go into like much detail. So we purified this enzyme because it's a human therapeutic drug. So we need to have very highly pure this drug. So we achieved almost like 98% of this purity of this drug. Then we checked its neurotoxicity by estimating this glutaminase activity. So as you can see that this molecule has significantly less glutaminase activity, which is responsible for the neurotoxicity. Then we checked antibody production in animals. So we took these two set of animals, and this one set of animals received this unmodified or the wild type asparaginase enzyme, and other set of mice, they received this M-ASPAR molecule which we have developed. And then we gave these three booster doses to this enzyme, and then to this mice. And then after 65 days, we checked this IgG and IgM antibody production in the mice. So as you can see, the mice which received this M-ASPAR molecule, which we have developed, showed significantly less production of IgG and IgM antibody, indicating that this enzyme is less immunogenic. The other property we're looking for is to have a molecule or the therapeutic molecule which does not bind to the pre-existing antibodies which are present in the patient. Because as I mentioned earlier, the leukemic patients, when they take this asparaginase drug, they produce antibodies in their body. So when these native or the commercially available asparaginase are given, these asparaginase, it binds to these antibodies and neutralizes. But what we found that when this M-ASPAR was given to the mice, so these antibodies did not bind to this M-ASPAR molecule. As you can see a data here, we took these primary ALL patients, and then we obtained this serum from these primary ALL patients, and then we checked binding of this wild type as well as our M-ASPAR molecule. Then as you can see, our M-ASPAR molecule binds significantly less to these IgG and IgM antibodies which are present in the patient body. And similarly, we found in the relapsed ALL patient. So overall, this data indicates that this M-ASPAR has a potential to improve this relapse ALL therapy. Then we checked pharmacokinetic properties of this enzyme. So we checked this half-life of this enzyme, that how long this molecule is circulating in the blood, how fast it is clearing from is cleared from the body, its bioabsorption, bioavailability. So we did this pharmacokinetic analysis. So for that, we administered the wild type asparaginase and our M-ASPAR molecule intravenously in the mice. And then we collected this blood at different time points, and then we checked for this blood concentration, half-life, exposure time, and elimination rate. So as you can see in the table, this AMASPAR molecule has better half-life, almost double. Then drug exposure time was almost double, and then it was cleared very slowly from the body. Okay, so this parameter shows that this AMASPAR molecule has better therapeutic or the pharmacokinetic properties as compared to the wild type enzyme. Uh, then we check that how efficiently this molecule can kill the cancerous cells present in the mice. So for this, we developed a xenograft ALL model. So what we did, so we took almost 10 million leukemic cells or the cancerous cells, and we administered these cancerous cells intravenously into the non-skid mice, and then we allowed these cancerous cells to multiply in animal body. So when this total leukemic cell percentage reached to 5% of this total cells, then that led to development of this leukemia or the cancer in animals. And then after that, we treated these mice with the wild type asparaginase and our M-aspar molecule. Then 2 more injections were given after 7 days. And then after that, we checked the survival of these mice as well as the percentage of killing of these leukemic cells in the mice. So as you can see in the efficacy graph, the mice which received this M-aspar molecule, there was significant killing of these cancerous cells in the mice. As you can see this red line as compared to this wild type this enzyme. And also we found that more than like 90% of these mice they survived when these mice were received our this M-aspar molecule, and the mice which received this wild type enzyme they died after 10 days. So this summarizes this slide summarizes this xenograft study. So the mice which received this wild type or native asparaginase, only 10% of these mice survived, and only 30% of these leukemic cells were killed by this wild type asparaginase. But when these mice received this M-aspar molecule, 90% of these mice they survived, and more than 80% of these cancerous cells were killed by this M-aspar molecule, indicating that this M-aspar molecule has a better therapeutic this efficacy. Then we also check, because when we are developing this any drug, it should not exert any toxicity on the vital organs like spleen, liver, brain, lungs. Okay, so we did a single dose and multiple dose toxicity because the cancerous patient has to take multiple doses of these chemotherapeutic drugs. So we mimic those conditions. So in one set, we gave single dose to the mice, and then we monitored these mice for 14 days. Then in multiple dose toxicity, we gave these doses after every 3 days up to 30 days. And then we checked the toxicity on various organs, also checked various blood parameters. I will not go into detail, but this slide just shows that the mice which received this M-aspar molecule did not show toxicity in any of these organs: liver, brain, heart, bone, spleen, lungs, and kidney. Whereas in the mice who received this wild type enzyme, they had like a toxic effect in some of these organs. So indicating that this molecule or this M-aspar molecule does not exert any toxic effect. So as I said, when you are developing these therapeutics or the vaccines, you need to have industry academia collaboration. So, and when we approach to the industries, they have very different thought process, because whatever we develop in the lab or whatever results we get in the lab, it's not most like in most of these cases, these results like don't get replicated at the industrial scale. So you need to like identify the industry who will be doing this preclinical and the clinical studies for your product. So we have now partnered with Tata Memorial Hospital and Actorate Mumbai, and also identified industrial partner DK. So this Tata Memorial Hospital will be like mainly doing these clinical trials, and the industrial partner will be doing this upscaling of this process, GMP manufacturing, and the formulation of this. So we have already done scaling up to 200-liter fermentation, and we got extremely good activity and yield. As you can see, like more 99% of this as per purity, and then activity we like achieved up to 10,000 international unit per liter, which is considered as a very high activity. And then now we also developed a formulation, and now three consistent clinical trial batches has been done, and now we are waiting for the drug controllers approval for to start this clinical trials. So this enzyme, what we think that can be a substitute for the existing other drugs, like the pegylated asparaginase, L-aspar, leucogenase, lunase, L-asparaginase, which are marketed in India. But how this product will be different from others, that it will be easily available, because this pegylated asparaginase, ervinase, these are mainly produced in Japan and USA. So they are not really available in India, and that's why many of these patients, they are not able to take these drugs. And also they are very expensive, because the pegylated enzyme is the treatment cost is about 120 to 140 thousand US dollars. So which is very expensive. So because of its like chemical modification, so our enzyme does not have any chemical modification, so it may reduce the treatment cost. And also our molecule has significantly reduced the side effects. Uh, and then it can also improve the treatment of relapse ALL, because it does not bind to the pre-existing the antibodies. Just quickly, I will go through the second part. So we are also developing the diagnostics for tuberculosis. So these are the like again various strategies used to develop the diagnostic these kits. So today I'm going to talk about the two kits which are under development. So this work is done in collaboration with our Japanese collaborator from KG University, Professor Biani, and then Professor Bigita from the to develop this TB RAP kit and ERICA kit. So why we need to develop this kit? Because as I mentioned in the beginning, that TB is a global burden. 1/3 of the world's population is infected with TB, and about like 10.4 million people, they are diagnosed with TB, and 1.3 to 1.8 million people, they die from TB every year. And as I already mentioned, that India accounts for 1/4 of these global TB cases, and um, so this is not like, as you can see, this is like a serious global problem. So this is a very like infectious disease. It is transmitted through the aerosols. So when this infected the person, or the person who is suffering from TB sneezes or coughs, it produces these droplets which contains this bacteria. And when these droplets are inhaled by healthy people, it goes to the lungs. Then in the lungs, this bacteria, it multiplies. And if it is not eliminated by our immune system, it then establishes the chronic infection, which leads to formation of the granuloma. And then once this bacteria remains in the granuloma, because it's a very rigid structure, the bacteria survives inside this granuloma. And when our immune system becomes weak, then it causes reactivation of the TB. So like it's very this bacteria has a multiple strategies to avoid killing by our immune system. So the most the challenging thing, or the high risk in the tuberculosis, is that about 35 to 40% of the TB cases, they remain undiagnosed, and that becomes a source of transmission of this disease, because as I said, this is highly contagious or the infectious disease, and if this patient, they remain undiagnosed, that becomes source of infection to other people. Another major challenge is that now there are multiple drug resistant, then extremely drug resistant, and totally drug resistant TB strains has been emerged, and these strains are causing a serious challenge worldwide. And to improve this diagnosis, then we need to deploy or develop some affordable, rapid, and sensitive TB cases, which can be deployed in rural areas. So currently there are many different types of diagnostic methods are used. For example, smear microscopy, then mycobacterial culture, chest X-ray, gene expert, which is molecular diagnostic method, then TrueNAT, LNA sequencing. However, these methods, they have several limitations, as you can see here. Like smear microscopy, it is less sensitive. Mycobacterial culture, it is time consuming. It takes 3 to 4 weeks to get these results. Chest X-ray is less specific. Gene expert, although it can detect this drug resistance, but it is expensive because it uses the cartridge, so it's expensive. It's TrueNat, like it requires skilled manpower. This LNA, sometimes it gives false positive. Sequencing, it requires infrastructure, and again, it is expensive. So these are like several limitations of these existing diagnostic methods. So what we need to do is to overcome the challenges. So we need to, and also some of these methods, these are invasive. So like the blood is taken, so that becomes an invasive process. So we need to develop a diagnostic which is non-invasive. For example, can we detect this TB, let's say from saliva or tongue swabs, which is called as the non-invasive. So can we develop a method which will be very rapid, can give us results in couple of hours, can be more affordable, sensitive, specific, which does not require skilled manpower, because it is like poor man's disease. Okay, so uh, uh, in primary healthcare setting, there are no skilled manpower. So can we develop assay which does not require a skilled manpower and also this infrastructure, so that these diagnosis methods can be deployed in low resource setting areas or in low and middle income countries. So, so we are collaborating with Professor Begita from Aarhus University. So we have developed this non-invasive TB RAP diagnostic kit. So we can diagnose this TB from saliva. So they have identified one biomarker enzyme which is produced by the live or the multiplying mycobacteria in the saliva. So we have developed a method to extract this enzyme from the saliva. So this is the flow chart of this assay. So the saliva is collected from the patients, and then this saliva, if the person is infected with TB, so it will have this bacteria, and this bacteria, once it lies, it will release this enzyme, and this enzyme, then the substrate is mixed with the substrate, which then amplifies or form this circle, and then these circles are conjugated with the biotinylated enzyme, which is then put on these LFS strips, and then you can get these lines if the person is positive. It's just like the pregnancy kit. You put the sample and then you will get this line. So on the same principle, we have developed this assay. So what we're trying to do is now to make it the one-pot reaction, because there are several steps are involved. So we're trying to make a one-pot reaction. So you just take this enzyme, add in one tube, and then you will get these results on this clip. And then also we are trying to develop some app which will like share this information with the hospitals and the patients. So we already tested this in a small number of these patient samples from this saliva and the tongue swabs, and as you can see that it shows a positive result in the saliva and tongue swabs. But of course, we need to optimize this assay to make it more specific and sensitive. But what we observed so far is that this assay works is a non-invasive. You can detect this TB from the saliva. It is very ultra sensitive because this enzyme, it makes multiple copies of this substrate, and then since this enzyme is produced only by this live bacteria, so you can detect only active TB. And the whole assay, it takes about like 3 hours, and it does not require any equipment, just like a freezer and heat block. So it can be low cost, simple, and since it does not require any infrastructure, so this can also be like deployed in rural settings. The other technology we are working on is in collaboration with Professor Manish from KG University, Japan. This is on the same principle, but this uses DNA and RNA, and this method will be mainly used for diagnosis of multi-drug resistant tuberculosis. So here again, it can be detected from the saliva. So we take the saliva, and then there is a nucleic acid extraction the key. Then once you extract this DNA, then it is amplified by a combination of this enzyme, which is then put on this strip, and then you can get this positive and negative results. And then again, we are developing this silent voice alert system for analysis of this data. So these are the different like enzymes used in this RICA technology, and they have developed technologies, they have made this PAIR, which stabilizes all, which like has the cocktail of this enzyme, and this can be stored at the room temperature up to 1 year. To make this system more automated, so we are also trying to develop a robotic system which will perform this assay automatically, because the TB samples are very infectious samples, so that puts the operator at the risk of getting exposed to this bacilli. So we're trying to mechanize or automate this assay by developing this system. So yeah, so this work, all these therapeutic molecules, development of therapeutic molecules and diagnostic is done in collaboration with several other groups, and there are many PhD students from our GRAP are involved in working on this project. So, and of course, this work is supported by various funding agencies, and this is done in collaboration with our industrial partners. So, thank you very much for your kind attention. Now, I'll be happy to take any questions if you have.
Thank you, sir. I thank from bottom of my heart for enlightening us on how we can mitigate the adverse effects of leukemia and tuberculosis. And for this stage is now open for the question and answer. If the participants have some questions, they can raise their hands. I mean, there is a hand symbol on this this interface. You may raise your hands so that we can allow you for the questions. Stage is now open for the participants.
They are in festive mode. No one wants to ask questions.
Probably yes. Sir, since nobody's asking, I have one question. There are few tuberculosis drugs already available. I mean, that collective name is DOTS, and that government every time advertise as well. So, what is your take on that?
Yeah, so there are a panel of like this. We call them as a first line and second line drugs. So these are okay. So these are like drugs which are commonly used for the treatment of this tuberculosis. But like, as I mentioned in one of the slide, uh, there is like emergence of this multi-drug resistant, extremely drug resistant, and totally drug resistant strains. And these strains, they have resistance. They have developed resistance to some of these first line as well as second line drugs. So, for example, treatment of extremely drug resistant and totally drug resistant tuberculosis is a big challenge, because in totally drug resistant, none of this drug is effective. They are not able to kill this. Yeah. So the DOT system is there, but like because of this multi-drug resistant TB, the use of these drugs is becoming a challenge.
Is it okay? Okay. This is one last request for the questions from the participants. Nagendra Singh, please unmute your mic and ask the question.
Yes, sir, am I audible?
Yes,
Yeah.
you are audible.
Yes, sir. So I wanted to ask that since the TB detection kit that you are developing, it's based on the biomarker method and identification of the biomarker, but it is like identifying the active state of tuberculosis. So if we want to, is it? I mean, my question is, can we identify biomarkers with the similar line, but for the latent tuberculosis as well? Like from
Yeah.
the saliva, let's say, for
Yeah.
example.
Yeah, so actually we are working on that. So far we have identified this biomarker for active TB and also for the multi-drug resistant TB. Of course, there are some biomarkers are already known for like drug resistant TB, but we are trying to identify like some new biomarkers. So we have developed both these kits based on the known markers, active as well as multi-drug resistant.
Okay, sir. Okay. Thank you.
Any other questions? Since there is no other question, I would like to thank our speaker, Professor Avinash Sonavane, for enlightening us with the um, uh, um, for the mitigation of the adverse effects of leukemia and tuberculosis, how we can fight back these diseases. Also, I thank all the participants here for making it a big success here. With this, let us conclude this webinar and hope we'll be bringing you all other topics as well. And keep visiting us on LinkedIn and other platforms. We have our website as well. So thank you, everyone. With this, we will conclude this webinar for today. Thank you, sir, for enlightening us.
Thank you very much. Yeah. Bye bye.
Thank you.